Good afternoon, everyone. I'm your host, Alok Kumaja. We would be starting the webinar shortly. Thank you for your patience. Good afternoon, webinar. Good afternoon, everyone. We are waiting for some more people to join. We will be starting the webinar in upcoming few minutes. I really appreciate your patience. Thank you.
Q&A session.
Good afternoon, everyone. We are going to wait for a couple of more minutes and then we will begin with the webinar. I really appreciate your patience. Thank you. Good afternoon, everyone. We would be beginning in another 1 minute. We are just waiting for a few more people to join. I really appreciate your patience. Thank you. Good afternoon, everyone. I hope you all are doing well. On behalf of Advent Informatics, I, Alok Kumarja, warmly welcome you to today's webinar on an exciting and highly relevant topic, Essentials of Drug Discovery, Small Molecule Docking, and Virtual Ligand Screening. We are delighted to have you with us and look forward to an insightful and interactive session. Before we delve into today's discussion, allow me to briefly introduce you to the organization behind this event, Advent Informatics Private Limited. Headquartered in Pune, Advent Informatics is a premier distributor of cutting-edge life science software solutions. Our expertise spans across chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, and management of chemical and biological data. We proudly serve a diverse clientele including leading pharmaceutical, chemical, and CRO companies across India, as well as prominent academic and government research institutions. At Advent, we are committed to empowering scientific innovations by making world-renowned software platforms accessible to Indian researchers, educators, and industry professionals. We currently represent a distinguished portfolio of global partners, including Molsoft LLC, Chemixon, KFT, Chempas, Certara, Transgen IT Solutions, and Keymodel. Our mission is to bridge the gap between scientific challenges and technological solutions, hence driving discovery forward. Now, it's my pleasure to introduce you to our speaker today, Mr. Aditya Mishra, Junior Application Scientist at Advent Informatics Private Limited. Mr. Mishra is a rising expert in field of computational chemistry, molecular modeling with a strong focus on drug discovery technologies. At Advent Informatics, he plays a pivotal role in training and supporting researchers in implementation of the software solutions related to virtual screening, molecular docking, and chemoinformatic workflows. His deep understanding in structure-based drug discovery combined with hands-on experience with industry-leading tools makes him an excellent guide for today's topic. Please join me in welcoming Mr. Aditya Mishra as he takes us through the essential of small molecule docking and virtual ligand screening, fundamental pillars in modern drug discovery. Mr. Mishra, the floor is yours.
Thank you so much, Alok, for a wonderful introduction. And very good afternoon, everyone present here. So, as you might know, I am Aditya Mishra. I am a junior application scientist with Advent Informatics. And today, for the webinar, we have chosen a topic which is itself, right? So let's go and begin today's webinar. Let me just recheck. Share my screen now. Is my screen visible, Alok? Can you confirm?
Yes, it is visible.
Okay, great. So firstly, I would like to introduce herself. We at Advent Informatics. So, Informatics was found back in 2016 by Mr. Sujit Sadhu, and we are the prominent distributor of molecular modeling, bioinformatics, and cheminformatic tools nationwide. We are also the first level of technical support what you all will be getting, what all our clients will get, and we are highly appreciated for our support as well. So, just last year in November, we had a user group meeting, and in that we did a little survey how good is our responsiveness towards their queries, and out of which we got 5 out of 5 stars. And also there was a question, how much time do we take to respond to their queries, and like half of them, 50% of them agreed that we are able to respond to their queries within an hour, and rest all agreed that we are able to do it within 24 hours. Also there was a question how good we are with solving their queries and technical questions, and out of that we get 4.8 stars, right? So we represent different softwares as well. So, for example, Chemexon is one of them, which is a global leader in chemical storage, chemical structure storage, chemical library enumeration, property calculation, and all these tools. We also are going to look into detail for the tool Molsoft. It is a complete molecular modeling suite. We represent Certer as well, specifically D360 solution. It is an integrated data visualization and analytical tool. If you're anyway into synthetic chemistry, medicinal chemistry, and all these things, you can definitely check out Compass. It is a AI-assisted lead discovery and forward reaction-based tool, right? So we have key module with it. Key module is a structure extractor, not just extractor. You can extract your structure in an editable format and from any kind of document, even the patents as well, right? So we are in partnership with Transgene IT Solution. It is a Bangalore-based company which is into ELN, compound data management tools, and all these things, right? So as per our topic today, we here are going to discuss about the drug discovery and development part, right? So the whole drug discovery and development flow starts with identifying a disease, right? You'll have to look for what are its proteomics, which amino acid or which residues are involved, what all things we need. We need to get a structure, get the whole thing about the protein, right? And then after, if we are able to do it, then we go and find a probable drug target that can be used to inhibit or either activate the specific target. So both of these steps takes times, and mostly, for example, it takes 2 to 5 years to isolate and discover all the things about the protein, and again it takes 2 to 5 years to find an effective drug against a specific protein. After we are done with this process, we do in vivo in vitro analysis, and we go for preclinical testing. That again takes time. If we are successful in preclinical testing, we then go for formulation and scale-up techniques, and during that we file an IND, which is intentional new drug application. And if that is a success, we go for human clinical trials. That again takes a lot of time, and if we are successful in this step as well, then we will get a approval from FDA, and we can launch our medicine for public use. Like, so the question rises how tools, in silico tools, are able to help in this, right? So as you might see, it takes a lot of time, and if we equip any in silico tools, molecular modeling tools, we can sort things out and chop off time in initial steps of drug discovery, right? So mostly molecular modeling and virtual screening tools are equipped for this. And like I said, different techniques are involved. Firstly, like if you're going to identify the disease and isolate the protein, you're going to need to study about genomics, proteomics, and biopharma relations of that protein. You're going to do high throughput screening of millions and billions of compound to get a good set of hit molecules. You're going to do a virtual screening to get best drug molecules out of that, right? And for that, we also use computational chemistry. We do molecular modeling, and at the end, we do in vitro and in silico ADME studies as well. So there are different techniques which are basically used during a drug discovery process. Firstly, let's say you can study about physiochemical properties and property calculation, like log P, PK. You can have a different kind of drug development approaches. Firstly, there is a ligand-based approach, and another is structure-based approach. In ligand-based approach, we have QSAR models, 2D, 3D, and everything. We can have pharmacophore model generation. You can, in a structure base, you have molecular docking, scoring functions, and also you can do de novo drug designing. Later on, we come to pharmacokinetic models where we see ADMET properties of our specific drug molecule or the molecules which is in test as of now. And at last, we can also equip cheminformatic tools. We can search through various databases which are available. We can do similarity or diversity search as well, right? So all these techniques composed together, we can count it as a virtual screening protocol, right? And the good part about today's webinar is we are going to look into a tool which is able to do all this, and that is Molsoft. Coming into the tool, Molsoft is a company that's just 31 years old, based on USA. It all began back in 1994 when the founder, Professor Rubin Abhigan, came up with two technologies. First is ICM, internal coordinate mechanics, and another is bias probability Monte Carlo method. Right? We are going to discuss about this in future and later slides. In the same decade, from 1994 to 2000. Molsoft got in collaboration with BMS and Genomics Institute, Novartis Research Foundation. In the next decade, from 2000 to 2009, we got some international recognition and got partnership with SGC for active RCM and scrap, and also with Novartis, we were able to get a new tool, which is ICM 3D ligand editor. Right. All these points, what you see in purple blue color, these are some of the success stories which represent how they used ICM to go through the whole process and were able to publish or find something out. And right, and in the same decade, we came up with a new technology, which is APF, atomic property field. It is kind of a pharmacophore model which can be utilized for other tools as well. Right. In the next era, next decade, from 2010 to 2019, while it was a comparatively new era for cloud computing, Molsoft was not behind, and they launched Molescreen and Giga Search engine, Giga Search in collaboration with Enum itself. And later in this decade, when their AI and machine learning is on a race, so Molsoft is not behind. Molsoft has launched RIDE, which is a rapid isostereoid discovery engine in collaboration with NCI. We have already introduced a new scoring function, which is RTCNN, that is trained on neural networks. Right. So there are different platforms which are using Molsoft as their primary CAD tool. Right. So let's go forward and look into the Molsoft users in India. So there are different reputed CAD CROs which are using Molsoft as of now. First of all, we have Syngene. It is a reputed name. Later on, we have Origin Oncology, Origin Pharmaceuticals, Zydus, Think Molecular, Eurofins, and all this. And in the academic side, reputed institution like VIT, Geetam University, Chanakya University, Naipa Guwahati, Bharatiya Vidyapit, Central University of Kerala, and the list is ongoing. All these are using Molsoft as their CAD tools. So let's talk about Molsoft. As an academician or a researcher, you might be wondering what is the scope of publication using the tool itself, right? If you just go and type the keyword Molsoft in Google Scholar, you will come up with more than 6,000 publications, and not just any publications. You can see there are reputed journals like Cell Death and Diseases, Nature publication, scientific reports, again Cell Death and Disease, and Cell. All these reputed publications do accept the studies which were done using Molsoft. So coming back into the theory part, the whole process of drug discovery consists of different steps, right? So there are steps where we do hit to lead approaches. Next, we do go for lead discovery. We go for lead optimization. Then after, we go to preclinical tests. And there are different tools which may be equipped to do so, right? These are the list which are generally these are the list of tools or techniques which are generally used. And with the use of Molsoft, you'll be able to achieve do all these things which I've just highlighted. Talking about the modules and functionalities of ICM itself. So Molsoft has a flagship product, which is ICM Pro. This tool we are going to use today for today's webinar itself, and we are going to do small molecule docking and virtual ligand screening with ICM Pro itself. So to do different things like VLS and homology, these are separate modules. ICM Chemist and ICM Chemist Pro are also separate module. But the good part is, if someone has ICM Pro with ICM VLS and homology, they will be able to do every thing what I've just discussed in the previous slide, right? So talking about the functionalities of ICM Pro itself. So as you might see in the screen, we have all different type of dockings with ICM Pro: small molecule docking, protein docking, induced fit docking. Inside induced fit docking, there are three different parts. We can do peptide docking, we can do RNA small molecule docking, we can do PROTEC modeling using ICM Pro. Also, we do have MD simulation with ICM Pro itself. So these are some of the snapshots out of it. This is a PROTEC model. What you say, these complexes, what you see here is the general graphical view what you'll be seeing. This is a snap from ICM Pocket Finder, and both of these are the part of MD simulation result. So there are some additional features as well. Firstly, you can definitely get a 2D ligand receptor interaction diagram. You can do homology modeling. You can do protein modeling. You can have biostatistic tools. You can have bioinformatic tools. You can also predict the effect of mutation. There is a separate algorithm for that. And the good part about the software is we do have different databases which are already integrated in the tool itself, in the interface itself. You do not need to go out of the interface, search for a protein, search for a molecule, and screen out randomly, right? So all these are definitely already incorporated in the interface itself. And these databases include PDB, Uniport, Protein Blast. For small molecule, if you want to go, we have PubMed, Campbell, Drug Centers, COD, crystallographic database, and all these things, right? So with ICM, we can see different type of pocket displays, right? So all the colors what we see in the screen are the different properties and properties of the surface, whether they are electrostatically negative, positively charged, either they have like a hydrophobic region, hydrophilic regions, and all these things, right? So you can have receptor pocket surface, you have ligand surface, you have electrostatic potential surface as well. So coming to the success stories part. So here I'm going to highlight a few cases where ICM tools were used. Right, so there was a publication from AstraZeneca where they can compare different tools to do post prediction and molecular docking. Here, as in the picture, you might see ICM was able to get the best results, and most of the poses is what ICM has predicted has lesser RMSD value than 2, which is compared very good as compared to other tools like Glide, Flexes, Goal, and everything, right? And also, all these tools, commercially available tools, were tested against different proteins, and out of which it was found out Glide was a choice of docking, and it gave a better result for 4 protein families, whereas ICM was a better choice and were able to give a better better results for 8 of the family classes, protein family classes, right? Also, it was found that ICM has a better enrichment factors as compared to other tools. So there is a blind industry-wide T3R challenge where a commercial as well as institutional softwares can participate, and they can see which is able to generate the best result as well, right? So there was Grand Challenge 2 back in 2017 where ICM participated, and ICM ranked first in binding energy prediction and RMSD for top scoring pose, and ICM was the only team where average RMSD was below 2 Armstrong, right? In the next challenge, Grand Challenge 3, things were different. This time we had 6 proteins to predict, and also this time we were able to rank first for docking pose and affinity prediction. Next time in Grand Challenge 4, the thing were different as well, where it was not about the smaller molecule, it was about the base macrocycline inhibitors, and again ICM was able to get top this competition in this area as well. So there is a lost platform which is mostly into oncology and immunological targets. They find out different drugs, and they use different techniques to do so, like PROTEC designing, hybrid activity molecule designing. They use ligand-based and structure-based approaches to optimize the molecules as well, right? So the key highlight is they are using ICM tools as their primary CAD tool, and in less than a year, they are able to get 3 molecules which have the activity of less than 1 nanomolar, and 4 molecules which have the activity less than 6 nanomolars, right? So if I talk about the success stories, there are plenty of them. I can spend the whole webinar talking about them, but this is the list. You can definitely go and check it out in the link below. And there is just a statement by Dr. Terry Roche, who is the head of computational chemistry in Lexicon Pharma. And the statement was, I had a tough case of tightly bound ligand in crystal structure whose bound pose were difficult to reproduce with other programs. They could not just find the right pose often. By using ICM, it fit right into the tight site and it gives terrific overlap over the crystal structure pose. So this statement we hear quite a lot with our clients, right? So talking about the VLS part, so back in 2013, like I said, it was a comparatively new era for computation, cloud computing. So what Novartis did, they undertook 30,000 cores from USA cloud computing, and they undertook 39 years worth of computation within 11 hours using ICM tools. And as a result, they were able to screen out 3 lead molecules, which are in trials right now. So, like I mentioned earlier, we had a group meeting last year in November, and these are some of the presentation what our current users has presented in the user group, and they shared how they are able to use ICM tools in their day-to-day research. For example, Dr. Srinivas from Shenzhen, he spoke about how he's using ICM tools not just for small molecule as well as antibody designing as well. Dr. Ramesh Sistala from Think Molecule presented about how he's using 4D docking and other tools in his day-to-day research. So you can definitely go and check it out in YouTube page of Advent Informatics. So going forward, let's talk about the technology what we have with Molsoft, how the things work, how Molsoft is able to deliver this precision. So firstly, like I mentioned, you might be wondering what ICM means. So ICM is based upon the technique which was previously in 1994, the founder himself came up with a paper where he used internal coordinate mechanics to plot where the atom is in a 3D space. So in general, all the other software which is available, they use Cartesian coordinate to determine the same. But in internal coordinates, we have different variables. The variables what we have in internal coordinates are bond length, bond angle, torsion angle, and phase dihedral angles. So in normal condition, all other variables stay constant. The only changes that happen in molecule are reflected with torsion angles. So by this way, we are able to give the precise results in the lesser times, right? And major application of this ICM internal coordinate mechanics lies for protein folding, protein modeling, molecular docking, and virtual screening as well. So there is another technique which is used to do sampling in ICM, which is bias probability Monte Carlo simulation, and it has 4 different steps. Firstly, the software will develop random conformers out of present molecule. Later on, it will minimize the energy of that molecule and in next step, it will evaluate all the additional energy terms like entropy, electrostatistical energy, and all these things. And if in the last step, it will select or it will accept or reject that conformer on the basis of overall energy, total energy. So there is a tool what we are going to demonstrate in this webinar, which is ICM Pocket Finder. Why it is important? Whether you protein or structure, what you have selected for your study, does have a co-crystal moiety or not. With the use of ICM Pocket Finder, you will be able to see what are the available cavities it has, right? Also, ICM Pocket Finder does has a scoring function, which is DLIT score, which was developed in collaboration with Merck. By the use of it, you can see how drug-like is your pocket, right? Talking about the scoring functions, ICM does have different scoring functions. Firstly, which have we have empirical physics-based score, which is ICM score. It comprises of all the molecular mechanic terms, their energies, and it is a addition of all those energy terms. Second, we have MF score, which is statistical knowledge-based scores based upon previous knowledge on the same. And last, we have RTCNN, which is neural network-based scores. It was trained from native ligand as well as decoy sets. So the good part is it does not use any other molecular mechanic terms. It is just based upon neural networking. So about physics-based scores, what I just mentioned earlier, there are different parameters which are considered. Firstly, we do check for the van der Waals interactions and internal energy field and field of the molecule. We look for free energy changes for the conformational energy loss. We look for hydrogen bonding interactions. We also undertake the size parameter. We look into hydrogen bond donor acceptor dissolution energy. We look into solvation electrostatic energy. We also have electrophobic free energy gain in the calculation itself. Like I said, RTCNN was trained on native like complexes as well as decoy sets, right? And there was an internal study by the team itself where they compared all the different scoring algorithms commercially or freely available, and out of which RTCNN was able to give the best prediction for top three poses. And also it was found in the same study that RTCNN score has better enrichment, this enrichment factor, that all the available scoring parameters, including our own ICM physics-based score. So there is another tool which is ICM Interactive 3D Ligand Editor. I'm going to uh, talk about it very briefly because we do have a webinar on this. You can definitely go and check it out on our website. This can be used to optimize our ligand in the pocket itself. You can do the changes in 3D and 2D as well, and see how that change or replacement affects the overall binding affinity in the terms of scoring. There is a small video, if I can show you with it. Like you see, I made a small replacement, and now the software is trying the replacement in the interface itself, and you can see it is trying for different poses, different conformations, and the best energy one, which has the least energy, will stay, and it will give me the score. Right. Let's go forward and see. So there is another technology which is APF, atomic property field. So it is a 3D pharmacophore potential map where it can be used for chemical superposition, screening, and scaffold hopping as well. It does have different parameters. So firstly, it considers hydrogen bond donors, hydrogen bond acceptors, sp3 hybridized states. It look for lipophilic regions. It does have size parameter. It looks for charges, positive, negative. Also, positive, electronegative interactions are considered here. So there was a publication in 2010 where it was mentioned that it is the most accurate method for chemical superposition and ligand-based 3D virtual screening compared to several other commercial or academically available softwares. Right. So this was mostly about the success stories and tools what we are going to use. And talking about the interface itself, let's see how the software use when we start with it. Whenever you open ICM window, firstly, it is very easy to use, and it is a choice of tool for biologists, chemists, and researchers worldwide, right? So whenever we open ICM interface, first thing you will see is a header part in which we have all the menus, all the sections which mostly we are going to use for our simulations. We have workspace pane on the left side, whether everything you take and import into your software that will be visualized there. You can toggle and not. You can use that to select the entities as well. On the center side, we have 3D graphical window. All the visualization is being presented here. On the bottom, we have a terminal. So terminal is basically the script what is going on in the backend, right? So advanced users do use terminal to work on the software. And also on the right side, we have selection button. You can select any entity using these buttons, and in the bottom side there is a tiny studio. You can make movies, you can create slides, you can take a snapshots using this window as well. So let's go and start today's webinar topic. And before that, let's talk about the different steps what we have in a general small molecule docking protocol. Like, we need to prepare the protein, we need to generate a grid, we are going to run the docking simulation, we are also going to analyze docking results on the basis of scores and interactions itself, and at the end we will validate our docking protocols as well, right? You might wonder, there is not a point where I'm going to prepare the ligand, because using ICM, like I mentioned in BPMC, Biosecurity Monte Carlo simulation, this BPMC used to sample and prepare ligand itself. We do not need to do it separately. So let's go and open ICM. Right. So this is how the software looks when you open it. Currently, you might see we only have two tabs here, right? We have the header section, we have workspace panel, we have terminal, we have 3D graphical window. And for today's webinar, the topic is small molecule docking and virtual ligand screening. So as you might know, to begin with a small molecule docking, you obviously need a structured receptor protein. You need a set of ligand or a ligand to dock into it, right? And with the use of ICM, you can definitely go and type in 4-letter code of the protein, because the search box here, like all the database are inbuilt in. For example, PDB is here, NCBI, BLAST, Uniport, everything is here. If I want to import a protein, I can just select PDB, type in the 4-letter code and import it. But that is one case. Another case where I do not have the idea about the specific the code of that 4-digit letter code of the protein, what I can do, I can just type in a family name, and I can see what are all the available structures here. For example, let's take in a family which is called CDK5. It is an impressive target when we look for like neurodisorders and all these things. So as soon as I typed in the family name, and either you can click on binocular or you can click enter, you will end up with a table on your screen where it has all the details what a general PDB web would have, like ID, experimental method, resolution, date, the title, the source organism, everything. Also, if we have a co-crystal molecule with the structure, it will be displayed on this mole column as well. You can definitely sort this table out on basis of, let's say if I want to see the latest structure, I can just go and set it into ascending or descending order. So the latest one is of 7/9/2021. Let's do this with the resolution, and now I have the least resolution structure on the top. So based on the table itself, you can select the structures. And to import in my software, I do not need to type in the whole code. Like again, for example, if I want to import this protein 1UNL, I can definitely go and type in the code and it will import. Or the easier option is just double click into any of the column in this table, and now the structure will be imported in my graphical window. I do not need this, so I'm going to delete this table now. Now, as you might see, 1UNL is now a part of my workspace panel, where I can see there are different chains A, B, D, E, and in A chain I do have a co-crystal ligand, and the same thing can be seen in my 3D graphical window, right? So for current study, what I'll do, I'll just keep chain A with which has an inhibitor in it, which is roscovitin, right? And I'm going to remove rest of the molecules because they are heteromultimers, right? So I'll delete this. So whatever I'm going to select in my workspace here is going to be highlighted in the 3D graphical window. For example, if I double click in my ligand molecule, roscovitin here, you might see it is now highlighted in green crosses, right? I can use my mouse scroll button wheel to zoom in, or either I can just select in anything and click on zoom, center button on the top side here, and now I can see there is a small molecule, a ligand in this pocket, right? Also, now you see it is represented in a ball and stick model, and the protein is represented in a ribbon model. If I want to change, I can just go to display. I can change the representation of the protein. For example, if I want to keep it in cylinders, I can do it. If I want to keep it in thick ribbons, and that is also an option. If I want to color it by secondary instruction, I can do that. Let's bring it back to smooth ribbon, right? And let's say if I want to see what are the interaction that my ligand molecule is making with my protein, right? For that, I'll just double click into this what I'll say entry, and I can go to pocket and ligand pocket interaction. But before that, you might see currently in my workspace panel, 1UNL is represented in XR structure, which is X-ray structure, and you can also see all the relevant data in the workspace as well. And if you want to look into the publication, primary publication, there is a PubMed reference just in this 1UNL site, right? So what I'll do, currently it is in XR form, which is in Cartesian coordinates. Like I mentioned earlier, ICM uses internal coordinates rather than Cartesian coordinates. So before doing the further study, I'm going to change it to ICM object. For that, I'll just go select this, right click. I have the option to convert PDB. As soon as I click into it, I have different options regarding what I need to do with the water molecules in it. I can keep the tight water molecules. I can delete all the water molecules. I can delete loose water molecules. So loose water molecules are those which are not in close proximity to either my ligand pocket or a protein surface, right? And I can also keep all the water molecules as well. So for this example, I'm going to delete all the water molecules because water molecules are not in very close proximity to my binding site as well. We have option to optimize hydrogen. Like I said, it is an X-ray structure. It does not have a hydrogen atoms as of now, and we are going to add in hydrogen atoms, and we are going to optimize hydrogens as well, right? We are going to replace the original structure with converted ICM structure. We are going to we have the option to hide missing side chains. I do not want to hide missing side chains. It is asking whether I want to predict the formal charges in ligand. So ICM does have a PK calculator with which can calculate the charges in the moieties. I'm going to optimize ligand tautomers as well. If there are tautomers in my ligand, we are going to assign secondary charges, secondary structures to our protein if there are not already assigned, right? And check on displaced results as well, right? So this is the only protein preparation part we will need to do. Rest all will be taken care by the software. And as soon as I clicked on OK, it took like 5 seconds and the process is completed. And if you want to see what are the changes that software had made, if you just elaborate this terminal part on top and scroll a little up, you might see the changes. For example, in this case, it has optimized 44 polar hydrogen. It has flipped different amino acids atoms as well, right? So you can definitely go and check the changes as well. So now, like I was talking about the ligand interactions. So for that, I just need to select my moiety, which is roscovitin, click right, go to pocket, select ligand pocket interactions. Now you might see our molecule roscovitin is making 2 hydrogen bonds. So the space what we see here, this is a blue one, this is a green one, this is a yellow one. So the size and color of these pairs represent the strength of our hydrogen bonds, right? Also, if you might see the neighboring residues which make the binding pocket are now labeled. Currently, they are in single letter code. If I want to change them in a, let's say 3-letter code, what I can do, I can just go into the toggle button, go to the label style and select, for example, 3-letter code, right? Now I have an idea about my binding pocket and what are all the available receptor residues around it, right? If I want to see the receptor pocket, I can again go into this, go to pocket, click into receptor pocket, and now I have the receptor pocket as well, right? So let's go and begin with the process where we are going to look into small molecule docking. For this study, what I'll do, rather than bringing up another ligand or a set of ligand, let's say I'm going to redock the current ligand in the same molecule and see if we are able to superimpose and mimic the natural co-crystal structure, right? For that, first thing I need to do is I need to make this site vacant, right? Already it has a molecule, roscovitin molecule inside it. First thing I'll need to do, I'm going to move this molecule out. So for that, I'll just go into my workspace panel, click right on my roscovitin, and I have the option for move from object. I'll just go and click into it. Now you see whether you see the roscovitin in the same pocket. Now it does not have the interactions, right? This means software considers this as a separate moiety, not the same one. And at the same time, in our workspace panel, now we have 2 different entries. One is 1UNL, which is our protein, and one is 1UNL1. Under this, we have our roscovitin. Another thing, we have a little arrow button here. If I click onto this, it will expand the sequence of my protein, right? And if I do this with the ligand moiety, I can see the 2D diagram of my molecule, right? So what I'll do, just I'm going to rename this ligand molecules as, let's say, roscovitin. And now I'm done with my protein preparation part, I do need a molecule to dock. So I'm going to redock this current roscovitin molecule, and I need the structure. I can definitely go and draw the structure, or either I can just type in the name and it will import the structure in my graphical window. I can save this in my as a table. But an easier option is I can just go into this and ligand enter in my workspace panel, click right, and I have the option to extract ligand. As soon as I click on extract ligand, it will ask whether I want to bring it as a just as a 2D diagram, or do I want to keep the 3D coordinates as well. For this example, I'm just going to import it as a 2D diagram. Right now, we have a new table called chem, and under this we have roscometin structure in it. So next step, what we are going to do, we are going to generate a grid for our docking simulation, right? So before looking into the docking part, let's see if this pocket is druggable. To do so, I'm going to go to tools, go to 3D predict, and I have the option called ICM pocket finder. I click on it, all the default parameters works well. Tolerance is currently 4.6. That is good for the molecules if the pockets are deep. If the pockets are shallow, you can decrease it to, like, for example, 2.5, 3, and all these things. I'm going to check. If you check on keep compounds, it will identify that there is a compound in your structure and you want to keep it, and it will not identify that one. And for this example, I'm going to keep it unchecked. Click on OK. And now, as you might see, we end up with a table where I have different tables which were ranked on the basis of D-lead, as well as different parameters. For the parameters which were considered are hydrophobicity, buriedness, aromatic, DLID, that score what I have mentioned in the previous presentation. So what I need to do, let's import any of these to this. I'm just going to click on the workspace panel. I can check multiple pockets at the same time and see where they are, right? And on the basis of this rank, it says the first pocket is best and most druggable. So if I click on this, this is the same pocket, UNL G pocket 1 UNL 1, right? So just to confirm, it has given the best score. And also, if I keep my co-crystal ligand in my working 3D graphical window, let me just make it a wireframe. Now, visually, it is also confirmed that our co-crystal ligand is a part of that, and it is deeply buried in this pocket. So it is a validation that this pocket can be used for our simulation, and grid can be generated around this. So this is one option, right? And to begin with the docking process, what I'm going to do, I'm going to go to docking, create a new project. Let me close this first. Go to docking, make a new project. You can rename your project. Let's say for this, I'm going to rename it as underscore 1UNL, which is my protein name. You can definitely select the folder where you want to keep your files. Currently, we have only one receptor object in our workspace, so it has already identified it. If you have multiple, you can just go and see in this drop down button, right? So it is next, it is asking for the graphical binding site residues. I can either select them with the use of the lasso button, what I have here. All the residues will be selected, and they are reflected in my setup receptor option. Or I do have the option to individually select my residues from workspace panel. If you just hold your control button and click on anything, multiple residues will be selected. The third option is definitely we can click on identify binding site. It will take us to the same ICM pocket finder option. If I click on this, see the same binding site will be reflected using this option, and it has now given me the best pocket here, which is 1UNLA, and I can use this pocket to do my to generate my grid. Or we do have another option where what we are going to do, we are going to use our co-crystal moiety in this case, which is roscoitin, double click into it, select it, and we are going to click on defined site around selected molecules, right? As soon as I click into it, all the neighboring residues are now selected. There are 28 residues which are selected, right? So after clicking this, I'll keep this checked, which is option of make receptor maps immediately, and I'll go click on OK. As soon as I click on OK, I have a green cloudy surface around my molecule, right? And also a yellowish blob, which is a probe which defines the reference point where to begin this docking simulation. It can be moved using your cursor button, but it is generally not suggested to do so. After that, I'm going to click on Go here, and now I have a grid which can be resized if I drag it holding any of the corners, right? And the size of grid can be definitely seen in the terminal window as well. So if I'm good with the current grid, and then I'm going to click on Go. So this is making the receptor maps where my docking simulation will run, right? Currently, we had like one was 1 UNL, another was just a second, let me, okay. One was 1 UNL under our workspace, one was a Roscoe moiety, and now we have third entry in our workspace, which is my doc underscore 1 UNL, whatever I named my docking project, right? So now we are ready to do our small molecule docking. We have our protein, we have a small molecule or a ligand molecule in it. I can definitely use a set of molecules. If we have, like, say, 100 molecules in a single SDF file, or I have to smiles of that, I can use in the same, right? So to go for docking, what I need to do is I'll just go to docking option, and I have the option to dock chemical table. I'm going to click on dock chemical table, and in our current project, we have two tables. One is pocket, one is chem. So automatically it identified that chem table is the one where we have the structure. If I keep it checked and click on OK, the docking simulation will start. But for you, let's uncheck this, and I'm going to show you some additional parameters as well. Right, as soon as I click on OK, now if I look into chem and settings, we have tools panel right here. It will open automatically if I keep that unchecked, right? So it will ask about the thoroughness of the simulation. So thoroughness is the effort of simulation what we are going to put in our docking simulation. If you have a flexible molecule and which have more number of rotatable bonds, it is suggested to increase this. Also, it will ask how many number of conformations do I need to run for this uh, data set, for example, small molecule, right? So by default it is 3. Let's change it to 5. It works well with 3 as well. So there are different options, whether I want to sample double bonds, whether I want to sample racemic, whether I want to sample tautomers, right? And CNN protocol is already equipped, so I just need to click on dock option, and in the bottom part you might see the docking simulation started. And see that in detail, I can just go to Windows, go to background jobs, and it will tell me the status of the job, the progress, the time, and also the output as well, right? Meanwhile, if we have any questions till now, you can definitely ask me.
Yes, Mr. Mishra. In fact, we have many questions coming in throughout the sessions. The question is, how can I see the length of hydrogen bonds?
Okay. Before answering that, so our docking simulation is done. So I'm just going to click on OK for this pop-up, and our results will be here. And to answer that, if you want to see the length of hydrogen bond, right? So let me include that part in our docking analysis, right? So firstly, as soon as we have our docking results, you will end up with a different table, which is chem docked, right? So the original name was chem, the doc table will be chem doc, right? Under this, we have 5 different confirmations here because I chose to get 5, and we have different columns here for score, number of atoms, number of flexible bonds. Like we mentioned in empirical physics based score, we have different parameters which are considered, which is hydrophobic groups, hydrogen bond energies, this van der Waal interactions, internal energy, dissolution, solvation energy, and all these terms. So score is physics based score, MF score is static scores, and RTCNN is your neural network score, right? So now if I check the top score pose, which is in rank 1, is given me the score of -38, and it has RTCN of CNN score of -49, right? So if I want to see it in my 3D graphical window, what I need to do, firstly, let me hide the co-crystal. I just need to click on the current pose, and now that will be imported on my 3D graphical window, right? If in this case, you might see you have hydrogen bonds as well, right? So if I go to this option where display hydrogen bonds, and I have the option to display the distance as well, right? So using this, you can show the distance as well. And what if I tell you we have another visual aspect? By the use of it, we'll be able to see it more clearly. To do that, I just click on setting button, go to tools, and we have a visualization panel, under which if I go to select pretty view and click on visualize. Okay, now let me switch off the CP Grace presentation. Close this. So I think this is a clear view where I can see the hydrogen bonding interactions. I can see the length. I can see all the different residues available. So the protein is currently is in wiry. I'm going to change it to smooth ribbon as well. And I can change transparency using light button. Now this looks much better, and you can definitely go to your studio part, make a snap out of it, and paste it anywhere you want. You can make a slide just clicking on this camera button, and the slide will be added on your workspace as well, right? So regarding the hydrogen bond query, these two techniques can be used. Also, there is another way by which we can see the top candidates. If I want to get into a conclusion which are the best poses, I can just plot a graph between two different scoring parameters, let's say physics-based scores and RT-CNN score, right? If I click on OK, now I end up with a graph plot on my window, and it has all the poses here. And if I see this quadrant where I have all the true positives, so if I select anything on here, that will be selected on my table as well, and I can import the same thing using a single click, right? So let's see how it performed as compared to our co-crystal ligand. To do so, I'm just going to import the co-crystal ligand, and now you might see our docked pose is very much superimposed over our co-crystal ligand without having any prior knowledge of the conformations as well. Right. So this is a visual aspect. If I want to see any statical aspect, so I can always go and check the RMSD of these. So for that, I'll go to tools. We have the option to analyze RMSD. We look for chemical RMSD. First molecule is our roscometin, which is co-crystal ligand, and another molecule is mydoc 1UNL receptor .m. So whatever you import from your table, any pose, that will be reflected with the entry molecule or M here under your this one mydoc receptor object, right? So these are both molecules which I want to check. If I click on OK, so current RMSD is 0.53. Any RMSD value below 2 is considered good, right? Also, if I want to see about the 2D interaction diagram of this, I can just go and right click into my docked pose, go to tools, and click on ligand 2D diagram. Okay, it will generate a 2D diagram of my current pose. You can definitely copy it. You can move the labels around by edit molecule option, right? And let's say I want to keep this whole project and I want to reuse it, the same project, to run let's say virtual ligand screening, right? So I'm just going to file, save project as. You can select the folder where you want to keep it. You can rename the project. You can save it. And let's say I want to use the same project and I want to run a VLS, which is virtual ligand screening. For that, what I need to do, firstly, I need a bigger set of ligand or small molecules. I can definitely use any SDF file which has the set of ligands, or in this case, let's do one thing. Molsoft ICM has a feature where I can see the ligand complexes from the same protein. For that, I am just going to click on mole screen and I'll go and see find Campbell PKD from mammalian data. So as soon as I click on it, there is a new table target from Uniport. If I find in the family name here, for here we use CDK5 family. Find it, and for CDK5 we have 587 molecules. So if I keep the box checked and click on get Campbell PKD data, it will import all the available small molecules along with their data, name, PKD, and their values as well, right? It has more than 500, like 657 molecules, so it is generally regularly updated. So to run for a VLS, what I need to do, I can just directly go to docking, go to set a batch ligand from SDF of mole. If I've already saved this file in my local machine, I can use this. I can index this database in my ICM interface itself, right? So to do so, I'm just going to show you how we can save it. I'm just going to click right on the header section and going to save it and keep this table as my for my virtual screening. I already have the same table. So what I'm going to do, I'm going to file, I'm going to open the same table what I have imported, right? It is the same table, and I'm going to go for docking, set batch ligand, browse in the same file. It has all the options regarding the ligand preparation. It will build hydrogen, it will assign charges, it will convert 2D to 3D. And if I click on proceed to launch, it will open my VLS, which is ligand screening part. And it will save my project name as my doc underscore 1UNL. Scoring docking effort is by default it is 1. We'll keep it that way and just click on OK. For this, I have a machine with the number of processes, so I can increase it, click on OK, and now as you might see in the bottom part, the VLS virtual ligand screening part is started. Like, do I need to wait the whole process to be completed to see my results? Not actually. Even if my process is completed, for example 7%, I can just go to docking, make a hit list, click on OK, and I can see how many of those 500, 600 molecules are docked yet, and I can score them. I can sort them on the basis of scores or RTCNN or whatever I want, right? So the same project file can be used again and again if you want to run on the same target, and you do not need to repeat the steps what we did in small molecule docking, right? So this was mostly about it. And talking about the presentation itself, regarding VLS, there could be various steps. Either you have the knowledge of your uh, structure, either you do not have the knowledge of your structure. That comes both in case of ligand or protein structure, right? So using ICM, there are different tools which can be utilized to run virtual ligand screening, right? So if we are going in ligand part, we have machine learning method as well as pharmacophore methods, and these are some of the tools what ICM has. Firstly, we have Bridge, which is rapid docking GPU engine. It is the fastest docking engine available in the market as per our knowledge. We have Giga Screen, which is uh, ultra large library screening tool, which uses Ridge as its docking engine. And we also have Ginger, which is lightning fast quality confirmer generator. Also, we have Mole Screen, which included all the 2D, 3D fingerprint pharmacophore models for different targets. There are more than 2,500 models of it, and by default, iSIM Pro has like 50 of those inbuilt. We have RIDE, which is Rapid Isostar Discovery Engine, that is based upon APF, Atomic Property Field itself, and it can screen about 1.5 million confirmers per second per GPU. And we are talking about a simple gaming GPU, not a very high-end GPU. We also have a Giga Screen that is effective to mine through billions of chemicals. Let's see the state of our VLS now. It is saying it has completed 7%. Let's go and remake our hit list. So now 48 compounds out of 657 compounds have been docked. Now, if I need to select a better library out of it, I can just sort it out of the scores. Or a good option is, again, I'm going to plot between both the scores. One is scores, one is RTCNN. I can color it as well. If I go to this color option, let's say I'll use MF score, my third scoring parameter. And now all the plots are here. If I want to select the best out of my available poses, I can just use this quadrant where they are both less in score as well as RTCNN. I can select these, copy these into a different table, right, and use this set to my following studies. So this was mostly about small molecule discovery, small molecule docking, and virtual ligand screening using ICM tools. Right. So again, we are here open for Q&A session. Before that, I would like to thank you for your attention throughout the workshop. And I think we are open for questions, Alok.
Mr. Mishra, the next question that I would like to ask is, what is the process to prepare ligands in ICM?
Like I said, we do not need to prepare ligands separately. It is generally processed on the fly while we are going to launch our docking the simulation.
The next question is, there are different scoring functions. Which one should I prefer?
Like I said, RTCNN has performed better than all the available scoring parameters commercially available or freely available, right? So RTCNN is good, and because it can also disqualify false positives. And always a better option is to plot between these different scoring parameters, like I just did, right? It will give you a better idea.
Okay. The final question would be, is it possible to run a molecular dynamic simulation with our dog complex?
Yes. If I have my complex saved in a ICM file, so I can just go into mole mechanics here, and we have the option to run MD simulation for the same complex. I can just select the object here. You will select the complex what you have, and give all the parameters, and you're good to go.
Okay. Thank you, everyone, for the wonderful questions coming through. Thank you so much, Mr. Mishra, for the informative and insightful presentation. Unfortunately, due to time constraints, we could not address all the questions submitted. But rest assured, all the unresponded questions would be reviewed, and our team will get back to you via email. with detailed answers. With that, we have come to the end of today's webinar. On behalf of Informatics, I sincerely thank Mr. Mishra for sharing his expertise and delivering such an insightful session. A big thank you for our audience for your engagement and questions. We look forward to hosting you in our upcoming events. Have a great day. Thank you.
Thank you so much, Alok. Thank you, everyone.